We're gonna witness the birth of one master student. We had one this morning, Maria.
Oh, good.
Maria finished this
nice,
morning.
very
Yeah,
nice.
that was very nice. She was very excited.
Good.
And now we're gonna have another one.
Wow.
Alan is kind of my son. He works with me in the lab. He always cooperates, and I hope he learned a lot of things in the lab.
For sure. Absolutely.
And now we're gonna listen to his research he did on the insulin receptor and neurodegeneration and the progression of Alzheimer's disease.
Thank you, Professor Alonso. I just want to say first my sincerest, heartfelt, warm greetings to everybody who made the time to come here. For Dr. Alejandra Alonso, I mean, she doesn't need introduction. Her description precedes her. And Dr. Sorry, I see you all the time.
Phillips.
Phillips. How are you doing? Dr. Phillips and Dr. McCluskey, who is also the chairman of the neuroscience department at CUNY Grad Center. And just want to special thanks to, actually I'm just going to cut straight to my mom who's here. No introductions needed. To Max, he's a former colleague here, and he came all the way from Brooklyn to be here to see this. Matt St. Arnold, my brother in the faith, and we stick to it like this. I've known him for years, so he's here to support. I don't know why this is his only off day, so he picks all days like this to come hear me. Like, I don't know, I think something's wrong with him. And then there's
Yeah.
Mr. and Mrs. Keeley. You have a common bond with them, actually.
Yes. He works with.
I worked with your husband,
Your husband.
with Dr. Bob, yeah. We met a couple of times, yeah.
And the Melendezes, we know very well.
Yeah.
Oh yeah, sure, yeah.
Yeah, it's nice to finally meet you.
Yeah.
Same here.
Thank you.
So they're reputation student too. They're another second set of parents. I have a lot of adopted parents. So between you and then the adopted parents and mentors and he took off his job to come here too. So he's another crazy one. So, I don't know, a room full of crazy people, I feel at home. So anyways, I just want to preface it that this is a culmination of research that I've been personally doing under the, of course, help of faculty members, including Dr. Alonso and many others that I've been able to pull from. Been doing an analysis for the past month or so, a long time, especially as I've been working in her lab, learned a lot, but then specifically in regards to this topic, been just researching, gathering a body of evidence together to present facts. Now, trying to present about this in this short amount of time is like trying to catch Niagara Falls in a bucket. But I will do my dead level best to take whatever knowledge, information that I've personally been able to collect up to this point and present it with the intention of just propagating that research on. It's a never-ending cycle, and as we all know, that it's especially in regards to disease like Alzheimer's, anything like that, there's always something needs to be done, and the more you know, the more you realize how much you still need to know about it. So with that, I'm just going to give basic introductions to the topic, and then we will just proceed on in time. And forgive me sometimes if I repeat something multiple times, but as I like to think, I'd rather repeat something multiple times than to not say it at all. So I just want to, hopefully, by the end of this, that something worthwhile will stick. So proceeding on, the purpose of this research is to demonstrate a correlation between insulin receptor activity and the risk or onset of Alzheimer's disease and related degenerative neuropathy. Now, specifically choosing Alzheimer's disease, there are a lot of different neurodegenerative diseases, but Alzheimer's is among the biggest in the field that we are researching to this date. and it has many implications for the future. And normally you would associate insulin receptor activity with diabetes, but in this research I hope to show this correlation. And now there's a null hypothesis to this, and there's an alternative hypothesis. The null, of course, being simply there is no correlation between insulin or insulin receptor activity in the brain and Alzheimer's disease. The alternative, which is what I would hope to present in this body of evidence gathered from scholarly peer-reviewed sources, is that there is indeed a statistically significant correlation between insulin receptor activity in the brain, and the onset and progression of Alzheimer's disease. So, drawing from this, now just a little bit of information just to get us started. It's the 6th leading cause of death in the US and the 5th among people in the 65 years and older age group. And the total estimated healthcare cost for this is estimated to be about $818 billion in 2015, and this number is steadily on the rise. And with a statistic that I didn't put here, but with a 2050 estimate that the world population would be 9 billion people by then, it's obviously significant, as that would imply both increase in the age of people living a lot longer, so you're seeing these symptoms which you would not have seen years ago when a lot of people were dying from other causes, like think Spanish flu, tuberculosis, you name it. So with that in place, it is imperative that large body research be done. Now I'm just going to lay out the presentation goals, just to have a different structure. So I want to introduce Alzheimer's disease, which we know a lot about it already, but I'm just going to just lay out the basic foundations on which we're going to build upon. And I'm going to introduce the significance of insulin in the brain. There are a lot of things that a lot of people might not know, that insulin in fact has this drastically different effect in the central nervous system than you would normally expect in the peripheral nervous system, and we'll get to that. I want to present analyses which correlate insulin and insulin receptor activity in the brain with Alzheimer's onset and progression. That's a big purpose of this whole presentation. Describe key critical biochemical signaling pathways of interest in the study. Now this is also too a very large bodied topic, but I want to just key in on the protein-protein interaction pathways that are specifically implicated in this process. And examine the correlation between insulin receptor and amyloid plaque. You'll see this come up multiple times throughout the course of the study. Build up with downstream. This right here, this amyloid plaque buildup and hyperphosphorylation of tau protein, these two things will come up very much in the topic. They are critical markers of Alzheimer's disease, so you'll see it throughout the whole presentation. And also too, I would like to, just a way of beefing up the database, introduce some experimental techniques and statistical mathematical analyses. Now, not all of these mathematical analyses, which I hope to cover in here, I could say I've done personally myself, but in the body of research in which these have been done, I would like to present statistical analysis, at least a good, reliable amount, and then connect it with why I believe that this can be used to correlate these two things with the statistical significance level, to show that indeed it can be implicated in a direct correlation. And then some criticisms and responses. There were a few papers that offered some little criticisms about this, to which I like to offer some responses. Some of them, 3 of which I can, 2 of which are more open-ended, but I like to show them anyways for the sake of just garnishing thought, and then just wrap it up. So, simple enough. So, now, Alzheimer's disease is characterized by amyloid beta plaques and tau neurofibrillary tangles. And these will be shown later on in the presentation. These are 2 huge things that's characterized by. And it's turned to be the largest cause of dementia, and also the risk factors. The 2 biggest that were presented are age and genetic predisposition. There are some genes which have been present, which show in populations that really drastically affect the onset of Alzheimer's disease. Others were environmental stressors, traumatic brain injury, other chronic depression, stress, psychosocial stress. Really could be think of as like a whole culmination of all these insults to the brain over your whole period of life, and eventually it just all just kind of like a wave that just comes up and then it manifests itself. So that's why I like to think of that when I put the correlation together. Because I like to get like the big picture of what's going on here. And without belaboring the point, so this is tau protein. Just for people who might not be fully aware of what it is, this is a major microtubule associated protein. It is critical in the brain for structure with tubulin. And I'll give an analogy so you can clearly see what it is. There are 6 isoforms in the brain. Here's the isoform. These bind to the microtubules. These are projection domains. And in your tau protein, there are many different sites where it can be phosphorylated. That will be a key. The key to this, though, is how it is phosphorylated, whether it's normal, because a good amount of phosphorylation is necessary and critical for biochemical processes. Too much, too little, as we know, a disbalance, and things go wrong. So I will show you that why this insulin receptor activity leads to this happening. Now I want to give a little so now we're going to do an analogy right here. Imagine in the brain an axon, a neuron. I want you to picture that the Staten Island harbor is a chemical synapse. I want you to picture the Staten Island train being an electrical synapse. I want you to picture St. George would be the nucleus of the neuron. The axon would be the railroad from St. George to Tottenville. The dendrites would be where the ferries dock on one side. So the ferries with people go across the chemical synapse in New York Harbor. They dock in the dendrites, which are the docks. They go to St. George Ferry Terminal, which is the nucleus. The train takes the people across the track to Tottenville, that's the axon, and then finally to the other side, and then it gets passed on. So I want you to picture that, but also too I want you to picture another thing. Picture that same example, but now we're going to illustrate give a very, very simple generation of tau protein that shows a more complicated picture. So picture that the train itself is what you would call dynein and kinesin. They're motor proteins. You can look on YouTube. There are all sorts of information about this. But I want to present they carry cargo along these microtubules. Tau is a microtubule associated protein, so it's involved in this. This is like a railroad track that keeps on reforming it wherever it needs to be. Like the neuron is just an amazing structure. It basically directs the railroad wherever the cargo needs to be at any given moment according to signals. So this is the train going that way, this is the train going that way. The cargo would be like the people on the train. Now imagine if your train's going along and there's a kink in the track all of a sudden. Guess what's going to happen to your train? Your train gets derailed, you're going to have service problems, you're going to have people posting on Twitter about how much they hate the MTA. So all that to say, this is a general picture of what I'm getting at. So this is this is a major effect of tau protein. So I just wanted you to put that in your mind as we go on. So tau is very, very essential. So roles of insulin. Now here's a part. We learned in biology class insulin is responsible for glucose uptake. As we know about diabetes, type 1 diabetes, a lack of insulin. Type 2 diabetes is insulin insensitivity, where the insulin receptors no longer respond to insulin. So instead of regulating the glucose levels, then you have all sorts of problems. You have hyperinsulinemia, all these problems. Now, insulin in the brain, surprisingly, this is one of the biggest facts that I learned, walked away with in this presentation, that it's drastically different in the central nervous system. Now, central nervous system is your brain and spinal cord, peripheral being the nerves in your appendages. So in the brain, it's been shown in different research articles, so transport across blood-brain barrier, it has implications of that, because nothing can just simply enter into it needs very controlled process, regulated. So it directly affects brain glucose level, indirectly. Now this is a key. It doesn't, in the peripheral nervous system, insulin causes glucose reuptake in the adipose tissues, in the skeletal muscle. Here in the brain, it does not directly do it. However, it affects the levels, because the insulin in the periphery comes over the blood-brain barrier because the brain is a highly metabolic organ. I've heard estimates like 20% of your body's metabolism goes to your brain. So all this glucose needs to go to your brain, goes across the blood-brain barrier, so insulin plays a role in that, and anything that happens in the periphery is going to go into central. So, and then that combined with any other, like just this A affects B affects C, it has an indirect effect, but not like the PNS, peripheral nervous system. And it's responsible for protein synthesis and cell growth, gene expression. In your brain all the time, genes are being expressed, depending on what is going on in your brain at that moment. So it's responsible in memory consolidation, especially in the hippocampus, where your memories are made and then they're stored in the neocortex. So the consolidation process, cognitive modulation, and in the amygdala, in the several areas, in the hypothalamus, and neuroprotection, kinase phosphatase. This is the part where the tau gets phosphorylated or dephosphorylated, meaning it adds a phosphate group. This adds a phosphate group, this takes away a phosphate group. So there should be a balance. If there's not a balance, things go wrong. And control of some people say apoptotic, some people say apoptotic, so you name it, but anti-apoptotic and apoptotic pathways, meaning apoptotic meaning some things need to get destroyed, a cell gets destroyed or cleaned up, and then anti-apoptotic stops that. So basically, this could be very critical in, say, cleanup mechanisms, where like something does not belong, it needs to get rid of it, or if something does belong. So basically, it's all about, really is a big controlling factor. Insulin is a big control magnetic process in the brain. And so now I'm going to present this quote. I'm going to make a bold statement here, and a lot of people criticize this topic, but that's why I directly quoted it. So see, and this is not just the only source that says this, but it says, although type 1 and type 2 diabetes are strong risk factors for AD, which they are, decreased insulin signaling in the Alzheimer brain typically occurs even in patients without systemic diabetes, prompting the suggestion that Alzheimer's disease represents a brain-specific or type 3 diabetes. And now I know some people might have a problem with this word, typically occurs, and once again that goes with experience in the field. I'm sure anyone who has experience in the field probably has a different perspective on it than I do. I'm just quoting what this source said. But there is now, starting in 1998, there was a journal by this guy, Hoyer, that kind of went towards this topic, but then in 2005, that's when an journal came out publicly suggest that Alzheimer's disease is not only comes on more readily with the presence of type 2, type 1 diabetes, specifically type 2 diabetes, but actually labeling Alzheimer's disease as a type 3 diabetes. And there are many reasons for that, include besides this, and there's just a lot of implications for that, which we can discuss later on. But basically, right now, this is what people are suggesting, and it's agreed upon by numerous body research too. So, and also could be the difference of the insulin receptors that plays into it. So now, I'm just going to show you some visualizations. We all like visualizations. So here is a diagram of type 2, type 1 diabetes. Here's your hyperphosphorylated tau protein. This pathway will come into play. This is part of a bigger pathway. And there's this GSK3 beta is active kinase, which is supposed to be balanced by phosphatases, which includes PP2A and a whole bunch of others. This balance should phosphorylate and dephosphorylate tau protein, but in this condition they're showing that it leads to downstream effects, which then causes tau hyperphosphorylation, which leads to these neurofibrillary tangles, which I mentioned as like the kinks in the railroad. The neurofibrillary tangles, it's like kind of like these tombstones. Now it's just inactive. It just leads to this degeneration cycle, and as purported, even as I remember purported by research by Dr. Alonso, that tau protein is shown to have a really central major effect on dementia, Alzheimer's related dementia. So this is important right here, and this is showing their amyloid plaque, which also as a result of the brain insulin resistance stemming from type 2 diabetes. So this is all affected. This is showing different genes analysis. The x-axis is Alzheimer's related genes, and then this axis is the insulin receptor related genes. This is showing some genes, yellow to green they are co-expressed. Green to light blue is that there's little or no correlation. Blue to dark blue are that the genes are anticorrelated. And as you can see, there is a certain amount of correlation between like the mTOR and the amyloid precursor protein. Over here you got some correlation. You have that apolipoprotein E, which is implicated in Alzheimer's disease. There's some there, some more over here with the, up here, with the insulin receptor substrate. So there's ample evidence there that there is a correlation between gene expression in regards to Alzheimer's and insulin resistance. And moving on, now these are just, I'm going to cover these pathways even more too. You'll see visualizations of it later on, but this is a pathway, the PI3K to PDK to AKT, or otherwise protein kinase B. This is a major pathway of concern regarding insulin receptor downstream pathways. And the Wnt and beta-catenin, which the Wnt is a major messaging molecule, especially present in early development, when your fetus is first developing. That has a major role in that. And then here's your two, this phosphorylates, this dephosphorylates tau protein, so a balance between those is necessary. And then you have other pathways that are reported, like mTOR, MAP kinase. These represent a whole bunch of downstream pathways from insulin receptor down to tau protein. So that's this. It's like a vertical aspect, if you want to think of it that way. So, and you'll see these visualized later on in the presentation. All right, so like right here. So here's your insulin right there, insulin receptor, and I'll just highlight the pathways of interest. There's your PDK, AKT, and PIP3 right there pathway, PIP2. This is a pathway I was shown before, which leads to downstream effects. You have, see, these are what normally insulin regulates. You have gene expression, glycogen synthesis, survival, and then you have a little bit of GLUT4. This is a glucose transporter. In the brain, the glucose transporters, for the most part, tend to be insulin independent, but this one, as you can see, is a little bit of influence there. And so you'll see this picture come up later on too in the presentation. And here's another one. There's your this is some receptors, some protein receptors, and then here's your downstream pathways, all leading to transcription of memory genes. It's like even when you're listening to me right now, this is happening in your brain. So this is the key critical role that insulin receptors are playing even now. They regulate this transcription process. And this is, I mean, it's a little bit pixelated, but you can see your PIP3 right there. This is that pathway right here that I'm talking about. And so here's what I was talking about before, the anti-apoptotic pathways. These are just the pathways listed. And the regulation of the Wnt signaling, which was discussed before. So basically this is showing the implication of insulin receptors. Now I touched on this briefly, the horizontal and vertical aspects. So I'm going to just build a basic picture for you to visualize this complicated process. So you have horizontal, what I noticed was horizontal and vertical aspects. And so you could think of is the horizontal being the result of insulin receptor and amyloid plaques, or amyloid, some they call them oligomers, like amyloid proteins that are resulting from this Alzheimer's condition. This is your plaques, your Alzheimer's, and your insulin receptors. You could see it as like a horizontal aspect right here. The vertical would be the insulin receptors and all these protein signaling cascades that descend from insulin receptor activity to tau protein. So you have your amyloid plaques, and once again, there are also more complicated in-process that go on, but just a general visualization right now, you have that, your amyloid, insulin receptors, tau protein. And so that will be shown in the next few slides. So here's the horizontal pathways. You have insulin affecting amyloid, you have amyloid affecting insulin. And some of the ways that I put here, a way that insulin affects amyloid, which remember, amyloid is a key component of Alzheimer's disease, just so we know where we're going with this. So studies have shown insulin stimulation accelerates trafficking of this amyloid beta from the trans-Golgi apparatus network, where it generates a plasma membrane. This is a major transshipment point, so it stimulates that process. And also shown to stimulate amyloid beta extracellular secretion and inhibits intracellular accumulation and degradation by insulin degrading enzyme. Now this enzyme is responsible for degrading both amyloid and insulin. So if you have a lot of one competing for the other, like say a lot of amyloid plaque, that could interfere with this enzyme degrading, or vice versa. So there's a competition over this right here. So additionally, a lack of apoptotic mechanisms regulated by insulin, which I had just been over, I showed you the apoptotic mechanisms, during insulin resistance could exacerbate pathological amyloid plaque buildup. So when you have a lack of these apoptotic mechanisms combined with the competition for this enzyme, then that could exacerbate the amyloid plaque buildup, which leads to other effects like inflammation, other stress-related effects, downstream, just all culminates. Now, you also have amyloid affecting insulin, including excess amyloid beta plaques compete with insulin, which I just mentioned before, and thought to reduce affinity of insulin to its own receptor. So it's kind of like, I picture like here's your receptor, here's your substrate binding, but you could picture like the amyloid getting in the way in a sense. So it inhibits that process, that according to what this research proposed. Now the amyloid beta, these oligomers, which I had just mentioned, inhibit neurotransmission of insulin-stimulated signals by desensitizing and reducing the surface expression of insulin receptors. This was also reported in research articles. And intracellular, this amyloid directly interferes with, once again, PI3K. This is phosphoinositol phosphate kinase, activation of AKT, and impaired survival signaling increased. Once again, here you go, this increased GSK3 beta, this glycogen synthase kinase. So once again, you have an increase of this, which you know what happens after that. It leads to tau hyperphosphorylation according to this body of research. So, and then on top of that, as some articles have shown, influence of microglia to release pro-inflammatory TNF-alpha. So basically it's like which I also show a demonstration of this, is that the amyloid negatively affects the microglia, which, as you know from immunological studies, microglia are a major immune system cell in the brain. It's like the monocytes in the peripheral. So you have the amyloid interacting with the microglia, releasing more inflammatory effects. Those inflammatory effects in turn affect the amyloid. So it's really this self-degenerating cycle. And so you will see, and which combined with, there's another one right there, J&K lead to reduced insulin responsiveness in neurons. So, okay, here's exactly what I was talking about. So picture, there's your amyloid beta, A beta for short. Now, when I was talking about stress, that could be, in a presentation I did before, I talked about psychosocial stress, which is like we typically think of when you think of like chronic, just malignant stress, just, you know, that constant depletive cycle. So that amyloid buildup right there is shown in this research article to negatively affect these microglia, which is a major defensive cell in your central nervous system. And as you can see, it leads to more effects, more stress-related effects due to inflammation, and pro-inflammatory mediators and neurotoxicity. So, and then once again, with a lack of cleanup mechanisms, which you would expect to be happening at the same time, it all just compounds on itself. So it's really one big exacerbating mechanism right here, downstream pathway. And so this is your vertical now. Talked about the horizontal. Your vertical is now both of these working together affect tau protein, which is the major component of this research, the tau, because that is where you get your dementia. That's where you get a lot of these biting phenotype mechanisms which you see in Alzheimer's disease, the deterioration, everything. So these A beta O's, amyloid beta oligomers, like little sequences, activate. Now here's another one, mTORC1. That's a key pathway. There's a few key pathways actually here, but there's the PIP3, PDK, AKT pathway. There's mTOR. There's another one that I'll show, which is the MAP kinase, and several that all interact together. And so they activate the mTORC1. Now this mTORC1 can be activated at the plasma membrane or lysosomes. Just to summarize the research of this article right here, plasma membrane activation of mTORC1 is considered to be detrimental. Lysosomal activation mTORC1 considered to be helpful according to that article research. Insulin can turn on both. Only amyloid can turn on that. So as you can see, amyloid leads to a detrimental, deleterious effect in stimulating this right here, which you don't want. And on top of that, there's been reported to be a balance because there's also mTORC2. mTORC1, mTORC2, and so the disbalance, disequilibrium between both of them is also part of the process. So you see a lot of this theme of this disbalance, disequilibrium of mechanisms that should really be effectively balancing out each other, but it's not happening. So now, insulin modulation of glucose. Now, I've mentioned this before, and what this is, as mentioned before, the insulin does not have the same effects on peripheral as the central, but taken into what it is, there are indirect effects, and the glucose, especially because there is a lack of glucose transport in this condition, which also, Professor, if I'm not mistaken, as you proposed, that the lack of glucose entering cells, especially with tau protein, could be a reason for extra hyperphosphorylation, because if the glucose is not binding, then that leaves more room for the phosphate. So obviously there's a lot of ramifications for this process, but basically that was reported in this article right here. And so moving on from that, so now I'm just doing a little collage. This is everything together, because we know we like to see the big picture, everything together. So just to make it all together, this is what you saw before, the downstream pathways from insulin leading to all these effects in the brain. You have here, once again, you have your insulin receptor right there, insulin receptor substrate, and then you have your amyloid precursor protein, which you have cleavage by these secretases. In this pathological process leads to a buildup of A beta, which leads to this downstream, which leads to neuroinflammation, as we discussed before, and then that leads to a bunch of further deleterious malignant effects. And then on top of that, here's your PI3K, and then to AKT, or protein kinase B, leading to increased activation of GSK3 beta, leading to your tau hyperphosphorylation. And then here's mitochondria. There's a lot of problems with the mitochondria in this relation aspect, in that they produce smaller amounts of ATP and give off more free radicals. So it's like a faulty car engine, where it's not producing the output it should at the expense of a lot more waste and pollution, and then which leads to oxidative stress. So with those mechanisms working concurrently, the oxidative stress, neuroinflammation, and there's some more downstream pathways right there. This is what I mentioned before. ERK is a major point where they come together. And then this picture, which I showed before too. So this is everything together. So up to this point, this is what the foundation that we've laid. Now, I'm going to go into some experimental designs. Now, for this part, what these experimental designs some of them are things that can be done short term. Some of them are processes that will require a lot longer term, a lot more careful analysis, and the proper equipment. But my goal, as I mentioned before in saying this, is I want to collect together a lot of different ways that this analysis can be precisely and sharply measured and built upon, because we need a lot of replicability in this research. As you will see a point I bring up later, a lot of these things are not that, they go back a ways, but they're not that old, if you will. So I just want to present some experimental designs, including mathematical analysis, that I would hope, if done correctly, would be able to have a fighting chance in further precise analysis. Now, first, I just wanted to go over, based on the body research, some things that can be done, especially in a laboratory. See this streptozotocin? That has been injected intracerebroventricularly into mice, rats, or you name it. And go figure, this STZ, for short, effectively replicates both diabetes and Alzheimer's in mice and rats. So if doing those experiments with this STZ injection, and then you further on do these other experiments with those mice, that is a perceived method to get more precise results. Secondly, which was depicted, experimentation has shown that injection of A-beta fibrils to the brains of those mice dramatically increased the number of neurofibroid tangles and neurons projected into the sites of A-beta injection. Additionally, and it was demonstrated that induction of insulin resistance in rats increased the A-beta production by increasing the expression of that secretase, simulating Y secretase activity. So, and there you have, once again, there's your IgE, your insulin degrading enzyme, which I covered before, degrades both insulin and amyloid. So when you have competition for that, especially if a lot of amyloid buildup and it competes, that's proposed effect for this downstream cycle. So now moving on from that, I just want to quickly go over, once again, I have a quote from a famous professor who happens to be sitting in this room. Yeah, things do stick in there. A picture is worth 1,000 words, a number is worth 1,000 pictures. So I was thinking, like, what's a good way? What can really beef up this analysis? And you know what? Truthfully, if done right, now we all always have bias, that's what we calculate for, but the numbers don't lie. So there are a lot of ways we can do quantitative analysis, statistical analysis, to find a correlation, especially being this is a correlational study. You might have heard the term statistical significance. Just going to briefly go over it. I don't want to go into too much of a rabbit trail, but this is pertinent to why I'm presenting this information. So there's quantifiability. If you want to get the correlation between A and B, you find what can I count from A, what can I count from B. I can do these statistical and analytical formulas, which I'll just briefly just show, and then see if there is a statistically significant correlation between the two. Well, the reason why I'm showing that, just a general synopsis. So if something is statistically significant, meaning if A directly affects B, you do the analysis, you would have a probability value that would fall here or there. This right here is a 95%, you can call it a confidence interval, standard error, measurements. Either way, if it was a perfect analysis, everything would fall right there. Obviously we don't. We have degrees of variation. This would be, what kind of variation can we expect around the average, assuming that there is no correlation? Like there's no like A and B are just, there's nothing there. Every single data value we got, it's just standard, you got maybe an outlier there, you got an outlier there, but it's all expected. But now if you do the statistical analysis on these two things and you find, oh wait a minute, my data value falls over there, and it's a two-way analysis because you can have greater or lesser than. Okay, this falls outside your confidence interval, especially you would use 99% if you're doing medical studies because you want to be as precise as possible. Then we can say that, oh wait a minute, something else is at work here. That would mean I would, in scientific terms, reject the null hypothesis and say, okay, there's something else working. And if your data value falls out here, like for instance, and I'll show you how this can be done in the study with insulin and Alzheimer's disease with the something I'll call biomarkers. Markers that, something quantifiable about this disease. That, wait a minute, this correlates statistically significantly, therefore we can see that there is indeed a direct correlation. So, now, first I'm going to show this study actually. This is an interesting study, and then I'm going to show this. So, this was done in China. There were 1,113 Han Chinese Alzheimer's patients, and then the same amount of control subject without added. Now you need both the same range of numbers. Now the Han Chinese population represents 90% of China, 19% of the global population. So the bigger your population is in significance, the more you expect that the effects there would carry on. Now, so in the study, which in my thesis paper, I have the actual graph in the paper, but what I wanted to specifically piece out from that was that in the study found that 336 out of 1,113 Alzheimer's patients had type 2 diabetes with standard deviation error range of 30.2 compared with 293 out of the same number clinically normal patients with, and there's a lesser range. And then notice it has a p-value of 0.048. So that's less than 0.05, which means it would fall outside of your variation you would expect under the null hypothesis. So assuming that this study was done right, assuming there was no bias, what have you going on, it seems to correlate along with everything else that there is indeed a statistically significant correlation between these Alzheimer's patients having these diabetes type 2 symptoms. And this, now the reason why I want to show this is I was talking about biomarkers. Now biomarkers are something quantifiable, and you can also, there's two things you can study with biomarkers, quantity and quality, because you can have, say, okay, disease happens, you have an increase of these biomarkers, or maybe they're found where they should not be, or you can have a qualitative, meaning something about the protein structure, the conformation analysis, something about its properties is different based on this. So now, in this graph right here, you have the clinical disease stage of Alzheimer's disease. You have your early, your cognitively normal. The Alzheimer's disease progresses before you actually see, like here you have, before you actually have your dementia and the mild cognitive impairment, because it goes cognitively normal, then mild cognitive impairment, then dementia. And you notice that even when a person seems to be cognitively normal, you still have a spike in your amyloid, your A-beta, and right there you have a spike in your tau-mediated neuronal injury and dysfunction. So there, right there off the bat, and then there's your brain structure already. So by the time you get to your mild cognitive impairment, you already have plaque, tau, hyperphosphorylated tau protein, you have brain structure anomalies, and then you have your memory problems, and then it leads to clinical function. So this is everything adding up. Now, I want to turn your attention to this. Here it says biomarker magnitude. Notice that they were able to project everything, the clinical disease stage, based on analysis of these biomarkers. So I want to hone in. The reason why I show this graph is I want to show how important those biomarkers could be. And I'm not even talking about, now let me preface this, for insulin activity in the brain. A biomarker could be anything that's directly or indirectly related. It could be one, the downstream proteins. It could be insulin itself. It could be glucose. Even though glucose would be kind of an indirect marker in this stage, it still can be considered a biomarker, as we know what happens to insulin deficiency and whatnot. And it could be, or you could go with, now Alzheimer's disease, biomarkers could include plaques, tau, hyperphosphorylated tau protein, or if you want, depending on the ramification of your study, you might include immune activity, you can include complement system, you can include glucocorticoid buildup, the stress hormones. You can include your TGF-beta, TNF-alpha, a whole interleukins, a whole bunch of things where, okay, I can take this piece right here, analyze and count and say, okay, this is normal, or wait, there's something else going on over here. Markers could even be something as, I'm going to inject this mouse with STZ, I'm going to make this mouse, which I will show you, I will make this mouse perform on a test. I'm going to score this mouse. If this falls within the score range, it's clear that this has certain condition, and I will show that. So I just want to show two tests that can be done. So I told you about the ICV injection of STZ, which basically reproduces this diabetes, reproduces Alzheimer's in that so far. This is the most that we have to date. There are certain tests you can do, performance tests. Morse water maze, for instance, where you give the mouse amount of time to find, like you put in the water, it goes around, and then you mark how long it takes to find the platform. And then you do it again. It'll show learning, like whether if the mouse is a very good learner, it will recognize, okay, it found the platform and go there faster next time. If something's wrong with it, then it'll just swim around and not be able to find it. And the Y maze is that the mouse explores. Like say it goes up that arm. And then now they like to search out novel things. So say a phenotypically normal mouse will probably go up that arm, then okay, next day it'll go up that arm, then this arm. But if you could tell something's wrong with the mouse if it's just, keeps on, they call it perseveration. It's like, perseveration is like say you have someone who they just cannot take their focus off of something, so you could tell if you were to, inject this with STZ and then replicate Alzheimer's, diabetes, you name it, and then put it on tests or like tests, there's more tests than this. You can score it, and then you could use even those results for a quantitative study. Remember, numerical. Okay, I'm going to quantitative study. So that's my reason for showing you that. Now, the next slide, I'm just going to briefly glance over some. Here we go. This is exactly what I was talking about before. I mentioned this before about the confidence interval. Potential biomarkers of insulin activity. You got insulin receptors, insulin, amyloid plaque. Depending on the ramifications of your study. Glucose transpose, IGF-1, IGF-2, Alzheimer's, amyloid, this p-tau is hyperphosphorylated tau. Immune function, and then the central nervous system stress. You can have glucocorticoids, you have your HPA, hypothalamus, pituitary gland, adrenal cortex. That's those three pieces that are really active during stress, and pro-inflammatory, anti-inflammatory cytokine ratios. So those are some quantifiable biomarkers as well as scores. And then this is just a list of different, I'm not going to belabor this point, but this is just different statistical tests you can do. The student's t-test, this is this analysis of variance. There's some tests to do if the data is does not conform to a normal configuration, which would be expected in a result like this. You got to be prepared for deviations from normality in your research. So this is the formulas. The Poisson distribution, which I want to show, is analyzing the distribution of data in relation to one another over a particular grid or area of interest. So say you have not only you're counting it, but then you see, okay, is there a direct correlation between wherever they are? You can tell that, if in a room, for us, if we're all sitting here and you say there's 5 people bunched up in the corner over there, and then there's no one over here, you can be like, wait a minute, maybe something's happening over there. Something is happening. So you're not only counting it, but you're also seeing in regards to each other, whether say this person is sitting over there and that person's sitting over there on the other room, like, something's going on between them. So it's showing both the quantity, but showing the way in which the quantity is directed with another, which can, when you consider the biomarkers of, say, insulin receptor activity and Alzheimer's disease, you can say, okay, these proteins, or whatever biomarkers you're counting, not only do they seem to be statistically significant number of deviation from what I would normally expect, but I noticed whether it's a lot of them in this area, and once again, you cut up the areas, whether it's digitally, bioinformatics, and you can see the relation one to another, and that could be a perceived method to see whether something is going on. And now, and this, you don't need to memorize that, that's just the Poisson distribution. And this also, I was thinking about, I don't want to go into too much of a rabbit trail with this, but at the same time, this is worth mentioning. In a wormhole that was opened up in a discussion with my father last week, that was basically on electrical engineering, we talked about this: Fourier transform analysis and statistical correlation studies using wavelength frequency. Once again, I feel like I'm trying to catch Niagara Falls with a bucket, but I'm going to do my best simply just to elucidate this in small terms, just simply to show why I believe if this is done with the appropriate equipment, I believe that this could lead to precise results in a correlational study such as this. So I want to highlight synchrotron. In this next few slides, just a few slides in this, I'm going to highlight this synchrotron radiation-based Fourier transform infrared microspectroscopy, excitation emission spectrums, reconstructing wavelength frequency, and biostatistical computation of quantity and quality of target structures for more precise data mining and mathematical correlation analysis. Now, I'm going to introduce, in the next slide, I'm just going to make an introduction of what it is, and then I'm going to show the correlation analysis right there. So, okay, so we all know the light spectrum. Here's your rainbow. We all seen a rainbow before. The rainbow is wavelength could be, sometimes you can apply to light, it could apply to sound waves traveling through the air. This example, you have a thin sliver section of light. Light is actually infinite. It's without end. Goes that way. There's no end to light spectrum. And it's funny, light's also a dual nature too, which is really interesting. But getting to the subject, so we only see with our eyes this small spectrum of light, but it's infinite. There's no end to the frequencies.
Excuse me, but there should be an end to the presentation.
Oh.
It should not be more than an hour.
All right. So basically, then I can. Oh, okay. So that's why she has a PhD. I know. so basically, my point saying that. So basically, the Fourier analysis. Here's your wavelength. The Fourier transform analysis plots this represents time right here. This is the frequency. This is the amplitude and frequency. What it does is it's going to, say, pass this through your sample. There's a Fourier transform and Fourier analysis, in which basically if it passes through something, it's going to break up, and then if you have a very sophisticated computer, it can piece it back together. When you pass this sample through something, whether the quantity or the quality is different, it will show up. If you have technology that was not new when the Jackson 5 were still singing, like if you have more updated technology computers, you can pass this. Sometimes, and different protein groups have their excitation emission spectrum. Different Eurochemistry teachers, you know this. That's where we work on with the confocal microscopy. And so, when you pass it through, it'll emit, it'll excite, it'll emit on the other side, and if you can analyze this precisely, then you can use this tool powerfully to look at mathematical correlations, and I'll show that in the next slide. Okay, so here, so we have your wave number, different chemical components. Now, the reason why I show this is because say if you have in your cascade of proteins, say insulin receptor, we know there's conformational changes. But if there's a chemical change, a biochemical change that occurs due to what's happening, you should be able to detect it with one of these wave numbers based on what it is. Now you know what the biomarkers are. So the Fourier transform analysis measured amplitude, I mentioned that, the vertical aspect and the horizontal. So you pass the frequency of the sample, you're testing for quantity and quality, and then the different compounds have different wave numbers. And this is also used in nuclear magnetic resonance spectroscopy, which also makes use of the wavelengths. So it's all the same idea, basically. And you use a computer to collect and interpret the data, and the Fourier analysis and synthesis is breaking it down. So as you go and break it down, you put it back together, and then with the computer programs that we have that are right bioinformatics, you can deduct, okay, because this is going on directly, this is why I'm seeing this. This is why so that's just a lot. That's a huge thing in just a few minutes, but that's basically what I want to show. And these are studies actually that have already used synchrotron. I'm just going to call it SR-FITR. In fact, this one right here is being used to detect nutrient deficiency in plant systems. Now, you know, sometimes the plants and specifically what they were looking for, once again, quantitative analysis. They were looking for calcium in particular. Calcium is enormously, phenomenally necessary in the brain because calcium is responsible for synaptic activity. There's nothing without the calcium. On top of it, just another little side note, plant also extremely utilize glucose, which is used in brain systems. So just those, but when I get it, it was used for a quantitative analysis, so it shows it can be used, and if used properly. And this one also, detection of unexpected fraud. So they screened, cassava starch, that's used in a lot of dishes in different countries. They were adding maleic acid to it, so they used the same thing to quantify whether this was present or not. So just going by that, if the same way is used to detect whatever's going on, the biomarkers that are present in this condition can be used. Now these are criticisms. There are three which I'm going to answer just right off the bat. Two are like, I can't really give an answer right now, but they are thought-provoking and lead to more research. So the one criticism rebuttal, okay, so the article said, only basal states of insulin signaling molecules have been tested, not their insulin-induced states. Now going by the cumulative research once again the professors probably have a different take on this with their experience. But what I was thinking when looking at this is that, first of all, studies conducted on downstream signaling cascade and the protein pathways would imply that these insulin-induced states are examined. I would like to imply, based on the data gathered in the examples up to this point, that in fact, that these insulin signaling molecules and their insulin-induced states have been looked at, because you would need to do that in order to say, okay, this is your correct phenotype, so how would I compare that compared to a diseased pathogenic phenotype? And knowledge about the effects and roles of insulin receptors in the brain, so would imply, so basically exactly similar to what I just said for the first one. The second one, okay, so discordant findings are reported in Alzheimer's disease brains on levels of insulin receptor and substrate level, and on basal activation states of AKT and G3K, 3 beta. Now, this came up a lot in what I was just talking about. So significant body of evidence, which from what I see, my perspective based on the cumulative research is significant, show a major role in that pathway on this activity right here. Even if the basal activation states of these proteins are not fully known, it is yet evident that this insulin receptor and insulin receptor substrate 1 play downstream effects on those pathways. Activation would lead to tau hyperphosphorylation. The numerous body of data which collected at this point do seem to indicate the direct correlation, and I do not believe, based on the findings at the point, that this would be a problem in the analysis of the correlation between diabetic condition, insulin receptor activity, and Alzheimer's disease in the brain. And I showed this to you before, just to repeat. By the way, it also that is geared toward mitochondria, by the way. So that's also, a lot of other effects. And now, I showed that to you before. This is just a repeat, so just kind of showing what I showed before. Now, there's evidence of reduced insulin signaling at the level of the insulin receptor, and and insulin receptor substrate 1, 2 remains to be reconciled with evidence of increased downstream signaling at levels of AKT, GSK3 beta, and more. Which, obviously, based on the studies to this point, I would venture to disagree with that statement. I think that it's true that there has been evidence of. Now, the point that got me though is I know there's differences, but it said increased downstream signaling. So that made a little bit. I'm like, does the downstream signaling get decreased or increased? Which that, of course, is a lot of evidence could be done into that. But I do know that there's a misbalance between this kinesin phosphatase and the GSK3 beta activity, hyperactivity is evidence of increased downstream signaling, because it's true that this is like a downstream, down effect of it. So it would be implied that if there's hyperactivity there, you would expect a reasonable amount of the downstream signaling to be. So, and then, so just closing discussion. So basically based on everything, obviously there's a lot more to be explored, but the results seem promising to indicate that a significant correlation between insulin receptor activity in the brain. Obviously insulin has all these really complicated roles in the brain, and so even though all the significance of downstream pathways might not be fully known at this time, I believe it is enough to induce that if any kind of progress is going to be made in Alzheimer's treatment, that it would be imperative to really focus on these insulin receptors in the brain, because they play such myriad roles that either way it would be imperative that something done with insulin receptors will either lead to a discovery or perhaps, which I'll show in the next slide, some research groups are looking at treatments actually. And there's more detailed information in my paper about these. I'll just highlight a few of them that stand out, like intranasal insulin administration. And this is one topic was looking at that, because you need ways to pass the blood-brain barrier. That's the thing. There's a lot of complications. So to deliver as efficient as possible. And this has actually improved cognitive defects in this research, and this was actually further reinforced by another article too, which talked about this. And then these also reduce GSK3 beta hyperactivity, which I mentioned before had an implication in the process. So all these are clinical application practicalities which should be considered for treating such a disorder. And so future areas of concentration, obviously we're not done. I mean, we're finishing now, but the research is ongoing. And I just point out some areas which I think really should be focused on. In vivo, first of all, in vivo insulin production. A lot of articles concur, obviously most of the insulin in the brain is shuttled over the blood-brain barrier. But there are some articles that do suggest at least a small amount of in vivo insulin production in the brain. So that's something that should be, of course, type 3 diabetes, which also, too, I can see, because think about it, between the articles and also, you have a lot of interesting between the peripheral nervous system and the central nervous system. You notice in the periphery you have a peripheral immune system, you have monocytes. The brain you have microglia. And even one article said the brain had its own renin-angiotensin system. And as you can see, the insulin receptors in the brain have a lot different function than they do in the periphery. So that would be evidence in the direction that, okay, this is, yes, it's very correlated with type 2 diabetes, but even more so, there's something very specific about the brain, which might not be able to lay a finger on, but it requires more study, basically. And then the pathways, of course, that's essential research and insulin receptors, and what I said before, the mathematical and computational analysis. We definitely should be looking to that. And then indirect, this is another thing, this has really baffled me, this has changed my whole viewpoint, because up to this moment I thought that insulin simply led to glucose reuptake in the brain, just as in the periphery. But that was all shattered when I saw, wait a minute, it's so much more than that. I mean, it has an indirect effect, as we could see, one into another, but it's so different. So I really would like to even see more research done with that process, and I think that would have ramifications for learning more about this disease. And I guess with that, I'm done. That's it.
Questions? We should have them, the questions from them first, and then we have our questions.
Not questions, but one of the things that was mentioned on there was using chromium percolinate for insulin resistance. My husband just took one because he's hypoglycemic, and so to eat the food, he uses that. So I think that that's pretty proven.
So we should ask questions, request them to leave us alone.
Yes.
I'm amazed.
You
Yeah.
should leave us alone with the candidate,
Okay.
and we will ask questions, private session.
Don't go too far, we shouldn't take more than.
Yeah, not more than 10 minutes. Let me see if I came, because
Sure.
she didn't come, yes.
Oh, I didn't see you.
No, she didn't.
Back home in Texas for 3 weeks, so he's doing great.
Hello, how are you?
Good job, Alan. There's a lot, lot of stuff.
A lot of stuff, yeah. Mental gymnastics. That's good stuff. I remembered when you did that class on the mathematical analysis, like quantitative.
Quantitative,
Yeah.
Stereology, yeah.
Yeah, oh, that's what's cool, yeah.
I'm just the opposite. I'm all about the pictures.
Pictures.
Forget about numbers. I know what numbers are.
So it correlates to both crowds.
It's true. You can see certain things in your picture.
Yeah. I kind of tend to be visual too, but I think having numbers on page has like a magical effect.
Depends on what you're trying to do.
Yeah.
Depends on your goals.
Oh, okay, we are alone. We are alone. Okay, now we should ask questions.
Okay, so you were talking about a horizontal and a vertical trajectory, and you don't need to. I don't know if I followed it 100%, but one of the ways that I'm thinking about it is that there could be many paths to Alzheimer's disease, right?
That's true.
So there's just like, I study epilepsy, and epilepsy is more than just one thing, right? There are many different ways. You can be born with a bad channel, you can abuse drugs and then quit them. There are many different ways that a car accident. But the end result of the seizure and the epilepsy problem is the same. Given all of your research, do you think that insulin, and I think you touched on a little bit of the criticism at the end, do you think that insulin explains all cases in Alzheimer's disease?
That's a good question. I would venture to say that it wouldn't explain all cases. I think, based on this research, it would explain a significant amount to the point where it's something that you should focus on more analytically.
do you think that, so type 1 diabetes is mainly genetic, right? Type 2 diabetes is mainly environmental. Which of those would you say type 3 diabetes is?
I would venture to say a combo of both, because it did say that the biggest risk factors for Alzheimer's disease were age, which I guess is, but your predisposition, your genetic predisposition is a big factor, but then on top of that, you also said all the stressors, say brain injury accumulation. I would say a mixture of both in a sense.
This drug STZ, what does it do?
So, that's a good question. So the streptozotocin, which it was, first of all, it was spoken about in numerous cases, numerous papers, and not just for Alzheimer's disease, but also other neurodegenerative disorders. So that would lend way to say that, yes, it is something that should be experimented with. What it does is, based on what I read, is that it just, especially if injected around the hippocampal area, it just mimics the degenerative effects. Now, I would have to research on it more to see what say if you inject a mouse with STZ, and now you see all these plaques, hyperphosphorylated tau. But my more straightforward answer would be it would just replicate that degenerated synapses and tissues, which is something you see in such conditions.
No. That's not a good answer.
Okay.
You have to find out there is a mechanism. STC, it has a definite mechanism, and it's used to generate basically diabetes. It's a model for diabetes. I think that interferes with the glucose metabolism.
So when you were talking about all these pathways to hypophosphorylated,
But you have to find out.
I was wondering if there have been any studies that, because we can detect phosphorylated tau, right? I was wondering, for example, if you add STZ, does it cause tau to be phosphorylated, or insulin? If you treat neurons with insulin, tau become phosphorylated? Or if you add A beta peptide, does tau become phosphorylated? Because you had arrows going from these things to tau. Have those things been done?
There were studies presented actually early on in the process with amyloid, and there does seem to be an indication in certain articles that can be done. It would definitely, once again, a key factor in this is the reproducibility factor, that at this stage it should be done more and then documented. But I would definitely venture to say you would see something along those lines where it's especially if you have a misbalance between the kinases and the phosphatases as a result of that. I would expect you to see such a morbidity or phenotype.
It makes me want to do an experiment.
No, but you should, I mean,
Absolutely.
this has been very much.
It's been done. That's why I figured that. I thought maybe there hasn't been.
No, especially with regards to the beta amyloid on the neurons have been done forever.
It kills them.
And they show, that they have shown that increased tau phosphorylation all the time.
Right.
I mean, that's the
Okay,
basis
right.
of the cascade.
That's
And
interesting.
they said that happened. I mean, the point is that it's very difficult to determine that that's really happening because of that and not because you're putting the neuron to a stress and it's I mean, it's very difficult, those kind of experiments, but it has been done,
Mhm.
and the results are very positive.
Mm.
Yeah.
So if you just kind of look at the neurons in a weird way, the tau becomes phosphorylated. I mean, it's very easily sort of triggered. Is that it?
Changes in tau phosphorylation is
Can happen.
difficult to happen, yeah.
It happens.
Yeah.
Hmm. Interesting.
You said that in ATP
Mm-hmm.
it gets depleted, right? I mean, it gets lowered. So how would you think, how would you speculate that tau gets hyperphosphorylated when ATP is lower?
That's a, so you're asking why is the case, it seems like kind of an irony that you have ATP. So, but what you're referring to is when I mentioned about the mitochondria producing less ATP at the expense of more oxidation. And on the one hand, you have tau protein being hyperphosphorylated. I would point those results to the fact of, first of all, in the mitochondria, we're talking about the whole adenosine triphosphate, and due to basically what is degradation of that, because when you have the electron transport chain,
Right?
so just a general degradation of that process would be expected to lead to reduced efficiency in producing. At the same point, for the tau protein you're talking about for GSK3 beta and the, say, PP2A phosphatases, you have a disbalance. If you have either a disc balance or the GSK3 beta is really active, you can see a mechanism where you have this G3SK that's out of control phosphorylating the tau protein, and at the same time you have a reduced efficiency of this mitochondrial electron transport chains, which is fewer ATP. So that is like the meat of the answer, combined with other, just related effects. On top of that, also I would consult the research that you had done, where you had mentioned about the glucose issue and hyperphosphorylation of tau in response to glucose deficiency. I would also consult that as a way to answer your question, combined with what I had mentioned.
Okay?
Okay.
Okay. So you come out, get out.
Okay. Steve.
All right. Well,
So we pass.
yeah.
Okay, good.
Yeah.
And then I will check the document. And I will not touch you.
Okay. Very good.
Sounds good?
That's good. Good.
Okay, good. So you have to sign on.
Sure. Do you have the thing, or he has it?
We have to get the forms.
The phones, paperwork, lots of paperwork.
I think so, yeah.
Good.
He's worried.
Yeah.
That's not good.
Okay. Good.
Okay, so we have to tell. Oh, here, he has the paper.
Oh, good. Yeah, I thought I saw it somewhere. All right. Ben?
Paperwork.
Well, he didn't fill it out. You gonna go here?
Yeah. Congratulations.
Oh.
You have some recommendations on your written document that you'll follow with Dr. Alonso, but the committee decided to pass you today, okay?
Thanks, Professor.
All right, good job.
Thank you.
Good job. You can collect the rest of your people.
Okay, I'll come up like bawling my eyes thinking.
You can take them first, take them first.
Do you have the phospho-tau antibodies?
Yeah,
You have
She's
all,
got all of them.
yeah.
The antibody for tau, everything.
Huh?
you can find all.
What I think is very interesting with this, because it's true that there is an influence on the insulin receptor, but I'm not so sure is that was causing the hyperphosphorylation of tau. You know, in the mouse, that's what the professor did once. In my transgenic mice,
Yeah.
there is an imbalance of the insulin
You did
receptor
it by
and the voltage sensitive channel. Yeah,
L-type,
sodium channel.
sodium channel.
Yeah. There is a different distribution.
Mm.
So, like, the hyperphosphorylation of tau could be controlling the level of the insulin receptor.
Mhm.
And maybe there is a crosstalk like that.
Yeah.
That would be very interesting.
Mm.
What did you put there?
There's, I mean, how many of these papers are there? It's
I
crazy.
don't know. How many we have to do?
It's like, I think
I
I
thought
just
there
bought
was
a
only
house.
one. What? I didn't know there was more.
There's three or four.
Oh no, because this
One is for
is
the written.
anticipated graduation date.
I was not supposed to sign that one.
That's okay. Yeah, this is the one that counts, I think.
There's that thing there's a thing at the Park Cafe.
Huh?
There's a thing at the Park Cafe today.
Yeah, we have to go to the park cafe.
Now, it's 2:30.
Yeah, we have to go.
What's going on there?
It's a celebration, let's go.
For what? For who?
For us.
For.
No, it's not for everybody, it's for us. People who do
You
things.
just got a nice
I
grant.
know, that's for you more than for us.
That's right, exactly. We just go to meet people who got big grants.
I just,
No, grants or papers.
I got my mid-tenure review article for our letter from Vivian here.
Oh, okay,
You read it?
okay. Oh, it's too low. Let's go.
Well, what are the outcomes of that? They're not-
What?
For the mid-tenure?
Did you get kicked out?
Yeah.
Yeah, he's gonna get kicked out.
I have to work more.
So you
Can
know.
you believe that she said she
To him?
she
Really?
found things to say.
Really?
Can you believe?
Didn't know what you
Of course, that's their job.
Yeah.
When we had Dean Park here, I told him something. He was like asking me about my work and life now. He's like, I know you have small kids at home. How is it? I said, it's a lot of work. He goes, I would suggest putting a cot in your lab.
That's what he said?
Allowed in now?
Yeah.
Don't ask me any questions.
Yes, we have you can You have? Oh, congratulations.
I was like, let me just tackle the stress and forget about all this.
After
Let me just,
Yeah.
let me just take that out of the equation, and then everything else.
Then there's this JC protein that exactly interfere with all the cascades going, just JC. Take care of that.
Yeah.
I'm thinking till, I was thinking, in fact, when he said the stress, I can relate to that stress, I'm like, maybe I need to like quit my job or something. 'Cause I'm gonna be like ADD and all that soon. What can I take?
I love that.
Pleasure control.
I love that.
And even if I end up with the big AZ, I know you can take care of it.
Well, you have to get focused. Yeah. Well, it's like, okay, these guys, this area, like, the line is right here, you know? And then other people have analysis outside, sociology, psychology, you know? And it's like, both of them affect each other. Here, and then you have political aspect, which kind of fits things up. But none of it.
I do, please.
You can't figure it out.
Yes, please help yourself right there. We have half and half sugars.
So it's like,
I don't think you're gonna open the fruit.
he might just come back and.
I don't think.
Really, how long is it?
Whatever.
There since like.
Then just, let me get the coffee so this gentleman can have a cup of coffee. You got that?
He's like,
Please help yourself to a cup of coffee, please, honey.
President.
yeah, there's the pretty thing.
Well, I didn't meet her in Mexico.
Oh,
He's survived. Oh, yeah, this is great.
Yeah, well, what Michael is in Cagayan at least is safer. It's those that are in a vegan. And like,
Like, they look weird. Yeah.
you just keep it like this.
What are the ones in the middle?
Oh, yeah, and you know what?
I'll be here.
Duarte,
I'll be here. We're gonna.
he's strong.
Obviously be there.
Of course, yeah, I'll be there.
Yes. You know what it is? When you're a
Yeah, it's
strong
nice.
leader, people love you.
I'm curious, 'cause my dad asked me that.
And,
So he
Yeah.
heard like this thing on NBC.
and actually, friends of mine know
Based on
Duarte
most
when he was the mayor.
doctor.
He was the mayor in
He said he
Development.
had this
So
internship
my
where he was
friends
making
know
like
him
6-digit
from that.
salaries. So my
He's
dad
very
asked me
good,
as,
so.
you know, nine members
You
of
get
that,
some plates, you.
Well, I showed you this
I can't think of anything offhand.
Oh.
That's something, just a
Yeah,
small
but please
thing, I
help
guess.
yourself. I got beverages.
Pharmaceutical company or something?
Yeah.
Could be.
Either way, I just felt like I.
Yeah. No, but
Thank you. Yeah, I appreciate you taking the time to. Of course, make
Help yourself. Let me get this. Oh,
Not
let
like
me
you're
get
sitting down,
some.
you know, watching TV all day.
That's not, that's,
I know it's a big thing.
no, it's good. It's always my pleasure to see good students and see it through and do what they need to do, so, great job.
And thank you for the visual analysis lecture, really stuck.
Good, yeah,
That's good.
Oh my goodness.
Please help yourself. I've got coffee, I've got seltzer, I've got lemon, I've got, thank you.
Thank you.
Thank you for
I'm
coming.
good.
Thank you. So good.
Oh,
Um,
yeah, mine.
I have the small plates too.
Oh my gosh.
Haven't done, so let me grab the small plates. I got cups, I got beverages.
Goodness. You got so much food here.
Like I said, my typical Italian.
Yes, well, I know that. Oh, here, put this down.
Yes, put that out
So it
too.
doesn't go to waste.
You know, that you don't want to go to waste, man. Put it right in the middle, right there.
Oh, here, tell me.
So friendly.
Yeah.
What school is that?
Thank you.
One in Canada.
There you go.
Right on the border of
Maybe I
What kind of
want to.
sandwiches now? Turkey?
Yep, it's an assortment.
And then I flew over.
I should have that.
And then I've got,
I actually asked John to come in.
Thank you.
here we go. This, beverages. I have iced
Oh,
tea.
thank you.
Like I said, you have, everybody from you to all the professors has been wonderful to Alan, and I'm very thankful.
And,
Mm-hmm.
Oh, he was a joy, believe me. He
Good,
was,
because I'd have to
he
beat
was
him.
one of our favorites, I have to say.
Thank you,
He really was,
thank you.
honestly.
I appreciate it.
One of our I'm sorry to see him leave.
Really is.
hey,
Maybe it's like his next step, which I keep telling him to network,
so,
is getting a job. That's his next step, and start going, you know, to pursue his doctorate.
yeah.
He wants to do it.
This is so bad,
What's the
right?
best
There's a little
best way to
Which is all right.
Well,
She, you know,
people here.
actually.
Well, that's what I told him to do. I said, network, network, network.
People just say, and then.
She'll have to lunch.
Yes, she needs to have some